Hey, cool. So thanks everyone for coming today. Yeah, my name is Brielle and I'm filling in for the branch organizers while they're having fun doing science in Rome. And I will begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes within the Suquamish, Tulalip, and Muckleshoot nations. And we also acknowledge that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So I'm happy to introduce our speaker today, Sophia Wassermann, and she is a postdoc scholar in the Punt Lab, working with the NOAA Northwest Fisheries Science Center on a project applying the CEATTL model. Yeah, all right, with a C instead of an S, to cannibalism in Pacific Hake with the NOAA Alaska Fisheries Science Center on a mixed species management strategy evaluation for groundfish in the Bering Sea and Aleutian Islands. Previously, she was a postdoc at University of California, Davis, working on portfolio effects and season timings in California fisheries. And she has a PhD from the National University of Ireland in Galloway, modeling the collective behavior of pelagic fishes to improve acoustic stock assessment methods. So Sophia will take questions.
Cool. I mean, if you pressing questions during, go for it. And there'll be plenty of time.
Yeah, and thanks for coming today. And we're excited to hear about your work. So my pleasure to turn it over to Sophia.
Thanks, Bree. And thank you all for being here, both in person and virtually. And also to the organizers of the Quant Sem, who I think are in Rome. So for them, for inviting me to give this talk. And so, yeah, I'm going to be talking about this work I'm doing, looking at cannibalism in Pacific Hake. And I've titled this talk, Cannibalism in Pacific Hake, Does It Matter? And I don't know if you know this law of journalism that says that any headline that ends in a question mark, the answer of the article is going to be no. But I'll leave it up to you to decide at the end of this talk if you think that it really matters. I think that comes from the idea that if the authors of the article could answer yes in the article, then they just have the statement as a statement. So I'm still unsure. I've moved this mouse around so many times. There we go. Okay. So fundamentally today, I'm going to be talking about ways to answer a variety of questions around climate-informed or ecosystem-based management, where the complexity of the ecosystem model isn't necessary, but where we think climate and trophic interactions are important for fully understanding population dynamics of a target species. And before I get too far into the models, I'll go over some of the acronyms. So yes, this is CEATTL that we're going to be talking about, the climate enhanced age-based model with temperature-specific trophic linkages and energetics. It's a great model. It's really hard to Google because if you put in Seattle, Google will think that you mean the city and change it to an S. And then that Seattle model is a model of intermediate complexity. So a mice model, and it's built around multi-species statistical captured age assessment, which is the SCAA, which is essentially an age structured population model for each species, which is linked through predation mortality. And the questions we can ask and answer with multi-species models are a little different from the single species stock assessments that are used for most fisheries. So here in the Northwest, as many of you know and have contributed to, we have very robust stock assessment models for single species, but there's always room to improve our understanding both of individual stocks and in their role within the ecosystem. And including predator-prey dynamics within these models can potentially reduce bias in stock status estimates, and then also improve the estimation of certain parameters, such as I'm going to be talking about time varied mortality. And additionally, CEATTL and other mice models incorporate climate drivers. And this is something that is increasingly important within stock assessment and within our understanding of ecosystems. how to include climate within these models, as we are experiencing the effects of anthropogenic climate change more and more. So this is one way to include climate drivers. And then it's possible to use models, especially in Seattle, within management strategy evaluations. So understanding how these multi-species models act within a management context and testing questions around that. And there's a paper by Grant Adams that will hopefully be coming out soon, specifically doing this if you're interested. And then also Seattle just fits well within the suite of models that we have to look at an ecosystem. So it's right between the single species models and then these more complex models and the system models like Atlantis. And I'm looking at Pacific hake, which is an incredibly abundant predator and prey item within the California current ecosystem. Supports a $60 million fishery as of 2017. And there are lots of trophic interactions that are important around hake. So it's an opportunistic predator. Its main prey items are euphausids or krill. And then also in the literature, there are estimates that range between 7 to 80% of their diet consisting of juvenile hake at different points, with that estimate sort of around 30%. And there have been demonstrated climate effects on their distributions and interest in exploring what climate means for their consumption rates. Also, this model starting looking at cannibalism is a great way to form a basis for more complex trophic interactions, maybe looking at adding prey species like krill or looking like at additional predators of interest within the ecosystem. And again, it sets up this basis for comparison between single species assessments and these more complex ecosystem models. So I'm using a package for this called RSeattle, which is a wrapper in R for the Seattle model built in TMB. So if you're comfortable with R but afraid of TMB, this is a great package for you. You don't even have to touch TMB. The package was written again by Grant Adams. It's all on GitHub. So if you have an interest, it's all available there with some simple examples to run through. And you put all the data into an Excel spreadsheet and plug it into this R package, and it'll plot stuff for you, super useful. And this has been used, the Seattle model and RSeattle, for a couple of systems in Alaska, Eastern Bering Sea, and the Gulf of Alaska. To run Seattle, you need data for an age-structured population model, a temperature time series, which informs the bioenergetics through a temperature-dependent consumption rate, and then also the diet data, which is the proportion of your prey species of interest by weight for each predator of interest within your model. And so I'm going to run through those data inputs now because I think it's important for understanding sort of all the different sources of information that go into these models. So first, for a structured population model, this is a time series of the population that follows cohorts through time with the parameters defined per age of fish. And these values were taken from the most recent Hake assessment and have included a time series of the size and the age of the fish, of the survey biomass, and then weighted age and selectivity for the fishery and for the survey, and also maturity at age, along with a bunch of other factors. And then within this model, annual recruitment and then the initial size structure or age structure are included as random effects. And for those of you a little less familiar with these sorts of models, essentially what they produce is a time series of the stock status, which then is used to inform the decision tables that are presented to the managers to determine how much catch is going to be suggested. So this is the basis of the assessment and what we can compare the outputs from Seattle to. And where Seattle differs from single species assessments is in mortality. So the model estimates the number of individuals for each species for this year or for the next year as a function of the estimate of the number of individuals of each age for this year times the mortality, which is composed of the residual mortality, which is M1, so that's from all sources other than predation and from fishing, and then M2, which is the predation mortality specifically for the predators that are included in the model, and then that's added to our fishing mortality. So for a traditional multi-species model, you would have multiple different types of predators and prey included. But again, for me, it's just the same underlying population dynamics for hake as predator and as the prey. And the predation mortality is equal to the amount of prey consumed divided by the total prey biomass. And that's roughly equivalent to our diet proportion times the predator biomass times this consumption ration. And so the consumption ration piece is where the next bit of the data inputs come in. So temperature is incorporated in two places within this model. First, because there haven't been a lot of studies of consumption rate within hake. It's used to estimate the optimal and maximal temperatures for this temperature-dependent consumption rate, which I'll talk about in a moment. Usually you'd have experimental studies to have determined what exactly those rates would be, but we don't have that for hake. And then you also need a time series with the mean temperature for each year, which actually sets that ration for the year. And there were two sources of temperature data that we looked into using. So first is the data from the acoustic trawl survey, which occurred for 13 years between 1995 and 2019. And we have temperatures reported for 100 meters or for the bottom if it's less than 100 meters. And these data in a study by Mike Malik et al. Were created across the survey area, which creates a spatial grid to which hake biomass was assigned. And then to get a sense of what temperatures the hake were actually found in, I weighted those created temperatures per spatial grid by the total, the estimate of hake biomass in that area. And I get a slightly different estimate of what the mean temperature was for that year. So those two lines are in purple and green, and you can see they're very similar, but they don't cover the entire time series that we're looking at. And if you have a missing year within Seattle, it'll just put in the mean value. So as the mean value for the entire time series. And so for the full time series, I also am using the outputs from the regional ocean modeling. Get what that S is. So summer ROMS values for the California current. And even when it's constrained just for the summer months, so the same months that the survey occurs, those values are a bit lower, which that's the orange line, but it's within about 1.5 degrees. So whether that would actually have an effect on the whole model, I think is potentially an interesting question, but not something I've gone too deep into. And so these temperature values are now plugged into this temperature dependent consumption curve. And so this equation is following on from previous instances of Seattle. So again, done in Alaska, maybe somewhat of a similar system. And then, so the consumption rate is modeled after this value from Kitchell et al. 1977. And so the plot shows the resulting rate per temperature. So you set, again, the optimal temperature and the maximum temperature. And this CQ value is this experimentally determined consumption rate value. And that has been determined for cod and Atlantic cod and for pollock. So those two values in the plot, cod is in purple and pollock is in orange. And we use the value for hake for the same CQ value as for pollock because we don't have one for hake. And I use the two different temperatures. So first, just the survey temperature, and then also this create value, which has a slightly lower maximum temperature. And they're very similar. There is a slight difference in the slope. So that blue line, which is this estimate from the maximum that I got from the create temperatures, is lower. But if you remember from the previous graph, the temperature series is around about 7 to 8 degrees. So that sits pretty well within our optimal temperature. So again, there are these slight differences, but in reality, I'm not sure how much of a difference it will make. And then the next equation for the bioenergetics is allometric mass, which is the change in consumption rate with the weight of the fish. So you have the weight of the fish on the x-axis here, and then the resulting rate on the y-axis. Again, we have the experimentally determined values for cod and for pollock, just for comparison. There is actually a study on the allometric mass for Pacific hake, which is from Francis in 1983. It's fun to use these papers from a while ago. It's not that common in fisheries that we're digging through them. And so he had two values. The first is represented in this green line. So this is for CA, which is the consumption by 1 gram fish at optimal temperature. And there was this experimentally determined value, which is 0.16 something. And then that was only during the feed and growing season, so he postulated that the true value would be about half of that. That's what I plotted in the blue line. And this gets really down into this nitty gritty and again, potentially important, but when it comes out in the wash across the whole model, I'm not sure it matters that much. And so we've used the original Francis values, that green line, very close to Pollock, again, would have been used in previous models for our bioenergetics. And so now on to the most important aspect of the data input, which is the diet. And this has been the source of the most frustration, but also the most interest for me in chasing down all of the data to go into this model. As I mentioned before, cannibalism is estimated to be anywhere between 7 to 80% of the Pacific hake diet. And in one study, there's a quote that cannibalism is of great importance to hake diet. So that's the idea that this project was predicated on. And so with that little scene set, I'll tell you the little story of my diet discoveries. So when I first started to look at the data, we were looking at the stomach data set from the acoustic trawl survey, which covers 9 years between 2005 and 2019. And this is a really awesome data set. It takes so much effort to collect all of these stomachs. There were almost 4,000 hake stomachs analyzed. So that's all of their contents, weighed and aged. And when I looked into the data, however, we found only 16 instances of cannibalism across the data set. So these are the total occurrences of all of the prey items found in this data set. And those tiny little orange bars at the bottom are our instances of hake. So that worked out to be about 0.4% of the hake diet being cannibalism. And so this had me a little worried. And I'm very glad that, you know, my continued employment was not based on whether or not. But fortunately, when we started looking at earlier data sets, we found records of cannibalism were much higher back in the 1990s. This is the same thought, but now expanded back to 1988 or 1988. So these data are from the California Current Trophic Database, CCTD, more acronyms. And that's a database of a whole bunch of different trophic studies that have been collated by some people at the Southwest Fisheries Science Center. And I do think it's interesting that while the rates are higher in the 1990s, and certainly more in line with studies suggesting that cannibalism was important for hake, this rate of occurrence of cannibalism still isn't extreme. And this is where I think it's important to note the difference for the diet data between the occurrence of a prey item and the weight of that prey item within the diet. So in terms of occurrence, this is from the CCTV. Pacific hake occur much less frequently than euphalcids. Again, that's krill, and even less frequently than some other fish and shrimp. But when we look by weight, hake are by far the highest contributor to diet within this database. And this makes sense given that your average krill is a lot smaller and weighs a lot less than your average hake. And it would make sense that krill would occur in much higher numbers than hake would. But I also think it's important to remember, this can be a bit misleading, that not every prey item was necessarily individually weighed. So those species of interest within the stomachs were more likely to have been weighed going back to the full data set and will show up as the highest contributors by weight. So it works out because Seattle uses the proportion in the stomach by weight, and I have the total weight of the stomach, so I can still get proportion of total weight, even if the comparison to the other prey items in the diet by weight isn't necessarily that easy to do. So it all works out. The talk doesn't stop now. There's no examples. And there was one more step in getting the diet data ready for Seattle. So since this CCTD contains diet samples from multiple different studies, we used a Dirichlet reweighting approach that's designed to work for combining data from multiple field studies. And I've used it, it's designed for multiple predator and prey species to be included, but I've done it for predators at age of hake and prey at age of hake. because that's what I'm looking at in Seattle. So the predators range between 1 and 15 years, and then the prey range between 1 and 5. And the diet proportion is first averaged across the predators to represent an average predator. And then the values are bootstrapped, which creates a distribution of diet proportions, including the uncertainty, which is then fit to a Dirichlet distribution, which results in a maximum likelihood estimate of the mean diet proportion for each predator and prey combination, including the uncertainty. So this graph on the right has each predator age in each box, and then the prey ages are these columns. And what's interesting to see, I think, is that the simple average, that's just the average before it's been put through this process, is incredibly similar to the bootstrapped average that we get out at the end. And that's expected with this method, but it does give us an idea of the uncertainty present within that average, which is represented by these 95% 95% intervals. And there's a lot more variance in this data set as they get older, represented by the variance across that time series. But this is what we have to go on. So the diet proportions are what I've fed into Seattle. So that's an overview of everything that's going into this model. Again, we have our assessment data, which is fed into this age-structured assessment. We have our temperature time series, which informs the temperature dependent consumption rate for the Hake bioenergetics. And then we have our predation, which again is the cannibalism. So now moving on to the results. And these are very preliminary. So I'm still playing with all the aspects of the models. There's grains of salt needed, isotope, tokens of frog, or whatever. So to start with, here are the resulting breakdown of the mean diet proportion by weight for the predator hakes. Those are the columns, and then the prey hake are the different colors. And when cannibalism does occur, so this is again averaged across that whole time series, it's mostly age 1 hake that are consumed. And the oldest hake consumed are age 5. So there aren't any records of hake being consumed older than that. And the amount of cannibalism increases with age and therefore with size, as expected, and sort of plateaus at about age 10. And you know with this axis that the maximum amount of cannibalism in this data set is getting up to about 15, up to maybe closer to 20%. So when that is plugged into Seattle, we can compare it both to the original assessment values and to the run with the single species without time varying mortality and without the predation element included. So these plots show the assessment in purple and then Seattle single species run in Seattle. If I can move that little box out of the way.
You can also minimize it.
Yeah. Well, I might show this. So the single species model is in orange, and then the model with cannibalism included is in green. And it's hard to see because they're very close together, especially for spawning stock biomass. But we see a little bit more of a difference with total biomass and increasingly more of a difference with recruitment. And that is likely due to the fact that it's mostly age 1 hake that are being consumed with cannibalism. So then when the model is run with this assumption of cannibalism, you get more young hake produced within the model. Also, we have more of a difference earlier on in the time series in the late 80s and the early 90s. And what I'm thinking right now is that there were big cohorts in 1980 and 1984, which resulted in, during that period, there being more big hake within the system who were then eating more of the youngest. If we assume the diet rations that I pulled from this data hold for that entire time period. So in general, this model with cannibalism does produce a higher number of youngest age 1 hake. And so another way to look at this is to look at the mortality. So the increase in age 1 hake is the product of increased predation mortality, which is M2 again, on those hake. So this plot shows the estimated natural mortality for each age of hake. So those are going to be the rows, and then each year is a column. And it's kind of hard to see, but there is some over here, a little bit of increased mortality on some of those hake up to age 5, which is again consistent with what we'd expect from the amount for the ages of the cannibalism that's occurring now. And so this model has a higher estimated mortality overall than the assessment model, and especially for these age 1 hake. And a benefit of running CEATTL is that we do get this time varying mortality. So you get a time series of what would be expected from predation specifically, instead of incorporating all into natural mortality. So not that much of an effect that we're seeing just from the amount of cannibalism that is created over this average time series. And so with that low amount of cannibalism, I thought it would be interesting to test what happens if you increase the amount of cannibalism and what would happen to the dynamics within the model. So what I did is run some sort of sensitivity tests on the amount of cannibalism that we put in. And so I have scaled the empirical value. So again, that reached a maximum of around 15%, to reach a maximum of 0.5% cannibalism by weight, 10%, 50%, and 75%. So I've tried a couple different ways of doing this, and I've decided that I really want to maintain that age structure. So this plot has, again, our predators in the boxes and our prey as columns. And the original empirical data is in the light yellow. And then as I scaled up the total amount of cannibalism, those are the other bars going to the dark purple. And when those values are fed into the model, we get this sort of rainbow effect of values coming out. So as the amount of cannibalism increases, we get higher estimates, especially for the total biomass and for the recruitment, which is to be expected again, because more of the youngest hake are being consumed through the cannibalism. And I do think it's interesting that for spawning stock biomass, there's not that much of an effect until we get up to the 75%. And then we do see a big jump in the estimated spawning stock biomass. And it's a similar story as before in the estimate of the natural mortality. And for these plots, I haven't made the scale the same, because if I did that, it would totally obliterate this again, this time series. So it's just note that those are not the same scales, and the total amount of mortality does increase when we get to the higher amounts of cannibalism. So that's again what we would expect. And I do think it's interesting that that time series does shift a little bit. The hotspots for the amount of mortality are different. And again, that's likely due to the underlying age structure of the population and how many of the bigger hake are around to eat the smaller hake, or how much the spikes in recruitment will change those dynamics. And so I think these sensitivity tests are a good way to explore the cannibalism dynamics, specifically the effects of these age structure in the diet data and what cannibalism means overall for our estimates of population. But I still have this big question around that time series. And so the reality of the variability within the data is that there appears to be this temporal trend. So this is the plot I showed you before of the occurrence of Pacific hake versus all other prey items within our diet data set. And these are from multiple different studies. So the amount of sampling has varied, and also the sampling protocols have varied. But I don't think sampling bias can completely explain why we're seeing this much variability over time. And so ideally, I'd fit time varying diet entirely, and you can do that within CEATTL, but any missing years are just considered that there would be no predation, which isn't likely to be true. So if anybody has a good idea of how to interpolate these years while preserving that age structure within the data, I'd be really interested to talk about different approaches. But for now, what I've been doing is I've fit 2 different periods of this Dirichlet related data. So on the top is the data that I just showed you fit into CEATTL. So that's for the entire time series. And again, that maxes out around 15%. And then I have a period for 1991 and 1999, which is up towards more like 80%. And then the most recent data, excuse me, which is for 2005 to 2019, again, those more recent survey estimates. And so when those are fit, I only ran Seattle for the years that were covered by those data points. So in these plots, the single species model is in purple. And then again, what I just showed you, that estimate for Seattle with all of the diet portion over all of the years is in orange. And then we have that earlier higher rate of cannibalism in green and the more recent estimate in blue. And this follows most of the expectations that, especially for this green data set, that first of all, there's more of a difference earlier in the time series in the estimates, and then also with the higher rate of cannibalism, it's going to lead to this sort of creation of hate within the model. It's also interesting to note that even though the cannibalism in the later years was lower, there's an interesting more of a difference for the model for just those years. And I think that has to do with the amount of years that are included in the time series. I only ran it for 2005 to 2015. That might be part of it. There might be other stuff going on. And I don't want to imply that this is actually time varying diet that I'm fitting here. I don't think you can assume that these curves continue for the little bit that's left out in the 2000s. But it's just another way to think about what has this change in diet meant in the past for this population, and starting to think about what that means in terms of the population now. So overall, my main takeaways have been that the occurrence of cannibalism appears to be highly variable, and that when that occurrence is averaged out across the time series, there is this small effect on the recent estimates of biomass and recruitment. There is more of an effect earlier on in the time series, which again may be due to the age composition, and that the estimates of natural mortality from Seattle are higher than the estimates for age 1 Hake that are included in the assessment. And then we also get this time varying mortality coming out of Seattle. And there's still a question for me around the importance that this has for the fishery. So the fishery selects age 2 and older. So given that the cannibalism is on age 1, does this not have that much of an effect on the stock that's actually targeted by the fishery? And then also, is it that important at the moment? So has the amount of cannibalism really dropped off? And if it was higher in the past, does that mean it could be higher in the future? And then what would happen for our populations? And I think the largest of the questions that remains for me, sort of existentially, and then also something potentially to look at, is this diet really varying in the past? And a full exploration of trends in cannibalism and hake diet overall is beyond the scope of my project. But just some interesting things, some interesting thoughts that I created as I was trying to think about this question is first of all thinking about some temporal trends. So first, is the sampling that's occurring now missing the instances of cannibalism that are out there in reality? So are they occurring further offshore, or are they occurring at a different time of year? So these plots show the frequency of occurrence of hake in stomachs for the sampling month for each year of sampling. And again, so from 2005 and later, those were all summer sampling during the Hake acoustic trawl survey. And it's possible that maybe there is more cannibalism later in the year. So if we look at 1997 and 1999, those are not months that would be covered by the Hake survey. And there is a little bit more cannibalism. This is purely anecdotal, but it is sort of an interesting question. Again, is it sampling, or has there been a change that's really occurred? And when we look spatially, so again, those years, 1997 and 1999, instances of cannibalism are potentially further south. Also in 1998, there was a huge amount of sampling up north. And then in 1995, it's occurring all over the coast. And there also doesn't seem to be that much of a trend in recent years, but again, there's just so few samples that included cannibalism. So again, I haven't done any analysis before beyond just sort of looking at this and thinking about this. But I do think it's important to keep in mind that hake are an opportunistic predator. They're not selecting juvenile hake out of preference or something else necessarily out of preference. So I think it's interesting to think about what kind of shifts may have occurred within this population that led to a shift in the amount of cannibalism, and what that means both in terms of ecosystem function and also in terms of hake dynamics themselves. So the remaining questions beyond the cannibalism time series are, how can this model be made to be more useful? So I mentioned that the fishery only selects at H2. So something that I'm going to do very soon is to run simulations of the CEATTL model at low recruitment. So we can look at what the combined effect of low recruitment and rates of cannibalism would be. So if recruitment's already low, is there going to be increased pressure from cannibalism? And then the next step is to look at adding complexity to this model. So again, this is a great basis for including more interactions. So there's interest in including krill as a prey species, looking at how changes in krill abundance may affect the hake population, and then also looking at what additional predators we can include in the model. So these are the most voracious hake predators included in this California current trophic database. And they're ordered by the frequency of hake predation relative to their overall abundance in the database. There's uneven sampling. And Pacific hake fall about halfway down this list. But we're interested potentially in looking at California sea lion, which is one of the most important. And there are some pros and cons of looking at sea lion. There is an assessment, that's great news. And hake do form a high proportion of their diet. So this is again the occurrence of prey species within the diet of the sea lions, and hake's right up there. But dealing with their diet would be slightly different and sort of interesting thinking about how that can be modeled, because the data are collected as otoliths within SCAT. So you can't get an age structure for the predators because you can't match the SCAT to the sea lion. And then also, while there are tens of thousands of records for sea lion, and they were collected all year round, which is really nice. They only occurred in 2 islands in California. That's this spot. It's San Nicolas and San Clemente Island. I don't know which one is which. I've never been to California. And they also, these records only represent foraging in shallow water, because again, sea lions have to have eaten something and made it back to land to leave the SCAT for the scientists to come and collect. So there are a couple of really interesting questions about how do you include these diets within the CEATTL model with the constraints of the population structure. But that's something interesting potentially for the future, requiring some creative thinking. And so lastly, I think this is cool and gives me a little bit of hope in terms of where this project is going. This is a plot of the frequency of occurrence of hake in the sea lion SCAT. So again, this is a time series now from 1980 to 2020 of the occurrence of all prey species, so again, purple, and then instances of hake predation in orange. And if you see, there is an increase in the 1990s in the amount of hake that were found in the sea lion SCAT. So that may be a suggestion that maybe there were more small hake in the 1990s, which were available both to the sea lions and to the larger hake. There might be some corroboration that there has been this shift in diet, which remains interesting. So with all of that, I'd like to thank all the people that I have worked with on this project. Thanks to Andre for being my supervisor, as well as everyone at the Northwest Science Center, especially Melissa. Thank you for coming. And also to Grant Adams, it's been a huge help with getting this project up and running. And to everyone else who's helped me with all of the data inputs. This has been a steep learning curve for me, and I could not have done it without all of these people. Yeah.
All right, so if there's any questions in person, we can have them, and also for people online, please put your questions in the Q&A, and I will read them out to
Is there any evidence for density dependent mortality with respect to cannibalism? Like, do they cannibalize more when there's more of them?
I would think that that would be likely, given that they're eating whatever they come across. I haven't specifically looked at that question, but I think there is some analysis that's left to be done, potentially looking at the time series of when these rates of high cannibalism are occurring, and what's going on specifically with the population dynamics.
Awesome talk. For the California sea lion hake otoliths, have you had a chance yet to look at if they're targeting the same age classes of hake as the few found in other hake?
So I know from talking with Alex Curtis, who handles the majority of this data, that it is smaller hake in general. At least that's my recollection. I haven't looked too deeply into it, but that is the assumption that they're relatively small and therefore relatively young. Yeah.
Have been any studies looking at the abundance of krill in the 90s at the time, so cannibalism was higher?
Yeah, I haven't stuck my nose into krill yet. I think that's another can of worms. That would be really interesting because, yeah, that's the assumption would be that if one prey source is more abundant, then they're shifting more towards that. Again, assuming there's no preference between, I think that would be mine, I guess. Yeah. So again, and these multi-species models allow you to look at those kinds of dynamics and in more detail and see what that would mean for the population.
Thanks for the talk. I really like how thorough your thought process was. I had a lot of questions and you kept answering them, so that's good. And I really appreciate the Halloween theme. Great job on that. But I'm wondering, do you think the spatial or seasonal model may be helpful for this too? And I'm thinking just how much the adult hake move across seasons. You know, a lot of them are down south where they might be overlapping when you fish in the winter, but in summer when you have most of your data, they're not. And do you think that would be helpful? But I realize you don't have much data.
Yeah, I think that would be helpful. And I think you exactly pointed upon why that might not be possible in terms of data availability. And also in terms of what we're trying to do with this model is a simplification. I mean, necessarily a simplification of these dynamics. And so I think my little exploration of those kinds of spatial dynamics is just to suggest that that's a really interesting question. And I do think it has a lot to do with the overlap in the different sizes and where and when that's occurring, and on top of whether or not we're detecting it.
Well, we have some questions online. First from Alberto Robolini. They say, great talk about the temperature effects on bioenergetics. Do you expect the CEATTL curve to be an important determinant of model dynamics, especially should this model be used to explore the warmer scenarios? And are you worried that using the same values of optimum temperatures for hake and pollock may lead to misleading dynamics, since these species may be expected to be acclimated to different temperatures?
Yeah,
that was long, but you can look at it if you want to.
Thanks, Algie. So yes, I think they are important. I do think that if that's a real point of concern, I think there's some sensitivity testing you can do around the bioenergetics. I have not had diet has been my main concern instead of the sort of temperature and bioenergetic aspects of the model. The CEATTL equation is actually, as far as I'm aware, better suited to warmer water species. So it's been used successfully for pollock. So I assume that it can be used successfully for hake, and that's the assumption we've gone on. I have used the optimal temperatures specifically for hake that are my best estimate from the survey. So again, we don't have the actual experimental values to back it up. I think the concern in terms of those values would be the optimal or the maximum, which ends up being pushed further and further to the left of the graph towards a lower temperature. And so if the temperatures within the time series are higher, then it would be expected that that consumption rate would fall off. For the temperatures that we're seeing that we're putting in at the moment, I don't think it's much of a concern. But in terms of thinking about this model under the context of climate change, then yes, the temperature becomes much more important. And it's another aspect that could definitely be tested within this framework.
We have another question from Rebecca Thomas. Since age 5 hake are almost as big as age 13, what's the thought for how that cannibalism occurs?
Yeah, I think that's interesting. I think it's interesting from a modeling standpoint in terms of having that age structure within the predation. I think you do kind of see that potentially if I go back. That the cannibalism does sort of level off as they get to be their maximum size. I am using ages specifically from the survey. So those are the age take, and then I'm using those ages to age the entire database. So I think that essentially it's just that the hake that are smaller are being eaten by hake that are bigger than them. And I think that's really where the cannibalism piece comes in. So yeah, only hake up to a certain size are being eaten because I don't think you can necessarily eat something that much bigger than you are. So yeah, that's my understanding of it.
We have another question from Elizabeth Phillips. They said, I think the California sea lion diet data could also be really interesting to look at seasonal consumption of hake. Perhaps we could learn more about hake in winter from their diet in terms of age, consumption, abundance, and distribution.
Yes, I think that is a really cool aspect. So the sea lion data are collected quarterly. So we have data for the whole year and sort of seasonality within that. And I agree that's a really awesome aspect of the database and a way we can learn about the hake dynamics during different points of the year. I think the only issue with that is again the constraints of the location. So again, we only have stat collected in these 2 locations. I've heard whispers of more data sets out there, so maybe that's a possibility. Maybe there's something further north. But still, even within those constraints, I do think it's a really interesting possibility.
That's it online.
I have a question you might not have the answer to. In your preparation of this project, have you heard or gotten the sense of at what point on like the management or monitoring side, like what's the threshold to get folks to fund another study or a longer time series of studies to look at hake stomach contents over the whole year? Like what's sort of like any hints of that? Anyone talk about that?
I think that is a difficult proposition because they're coming from this survey. There may be other routes for collecting stomach data, but I think it's. I don't know much about the budgetary or funding, but the stomach contents analysis is a difficult and time intensive process. So yeah, I mean, I think you can potentially get it from the commercial catches, but I think there are probably questions about how to make sure that those data are, can you get the stomachs? How can you make sure they're matched to the hake they came from, et cetera. So I think it would be really interesting. And then, I mean, if I had money just to throw at things, I think that would be a really interesting thing to do.
I'll jump in and say that this sounds like a cooperative research project with industry. And that's how you get money to do it, because we're not gonna get any more shit. Yeah.
So when you have, a job where you have money to throw, let me know.
Yeah.
What's the observer coverage like on these boats? Can they not just collect stomachs from the observers?
Maybe.
Is it 100% coverage? The commercial? And then-
Maybe remember.
I think at the FC it is, but, the shore side. They do port sampling. You could potentially, the problem with that, the fishery only operates May to November,
They're still not getting.
so you're still not getting that real critical time when it could be a lot over.
And that's actually, it's an interesting point, because I imagine, I haven't looked deeply into where all these sources are from. But if you look at the months where the sampling occurred, there's not much past May to November. So that's another hole that's within this.
They did a winter survey a few years ago. I'm not sure if they collected summits on that. It's like, what, 5 or 6 years?
Yes. Andy Parker Setter ran that. Beth and Julia and company was now. Great.
Cool. Thanks again, Sophia. Yeah, those are great.